So today I'm going to be talking about aspects of HIV. It is sort of going to be, tricky for me because I have a lot of stuff that I'm going to be trying to fit into one class period. I am probably not going to get through the slides I want to get through today, and I'll just leave heartbroken. Because as an HIV researcher, this is stuff I really enjoy talking about. So hopefully I'll get to what I want to get to. If I realize I'm getting to the end and there's some, I will mention sort of, hey guys, getting towards the end, are there specific things you want to know? So I can make sure to answer whatever questions you have, although I will sort of start from the beginning. This is a paragraph taken from the first ever report of AIDS. It was written in the CDC's Morbidity and Mortality Weekly Report, and which is basically a report of different infectious diseases in the country. Usually it's pretty sort of dry, like 4 people had monkeypox. Nobody had monkeypox in the US this week. But so here it says, in the period of October 1980 to May 81, 5 young men, all active homosexuals, were treated for biopsy-confirmed pneumocystis pneumonia at 3 hospitals in LA. 2 of them died. All of them had previous or current cytomegalovirus infection and candidal mucosal infection, which is a yeast infection. So this is in June 81 that this was published. In July, there was a similar report published about a group of patients in New York who all had some weird cancers. And if you actually go and read this report, it has a little bit of kind of editorializing at the end. So usually they're really dry, but at the end it's like, the fact that these people all were previously healthy and fine, and they all got these weird diseases that usually we see only in old people or people who are immunocompromised, makes us think that maybe there's some new disease around that is leading to people being immunocompromised. And that was sort of the end of it. This disease eventually became named AIDS, or acquired immunodeficiency syndrome. Patients who have AIDS generally die, or not generally, pretty much always die of infections other than the AIDS diagnosis. So basically what's happening in these patients is that these patients are all immunosuppressed or immunocompromised, so that other types of infections that most people would be able to fight off actually lead to their death. And so these infections are known as opportunistic infections. They are taking the opportunity to cause damage in these patients. The most famous ones are pneumocystis pneumonia, which was mentioned in the previous slide. This cancer that's caused by a virus called Kaposi sarcoma that leads to some spots like this. Whenever I watch an HIV movie, of which there are many, sometimes the person in the movie is like, oh, I have a weird thing on my neck. They always have like this drawn purple thing, and I'm like, oh, that's the Kaposi's. Sometimes they'll have some yeast infections like this, but there are a huge number of potential opportunistic infections that can cause problems in those patients. One thing that is mentioned here is Mycobacterium tuberculosis. Tuberculosis infections are actually kind of a major complication worldwide in AIDS patients, and so the tuberculosis and HIV co-infection issue is a big issue. There are also other types of tumors, and one thing that's not listed here that we are seeing more and more often is neurologic disease or neurodegenerative disease in some long-term HIV patients. So the reason why these AIDS patients are immunocompromised is that they are specifically suffering from a loss of CD4 positive T cells. So what you can see here is that CD4 positive T cells start at some high level in most people. The actual number that most people have at rest does vary, so this is sort of an average thing. People will eventually, as they are AIDS patients, see this decline in CD4 T cells. once their CD4 T cells get below a certain level, they start to come down with different opportunistic infections. The definition actually of when you have AIDS has changed a bit over time. It used to be when you had an opportunistic infection, that was AIDS. Now we're pretty good at doing something about opportunistic infections, and so instead we define AIDS based on your CD4 count. And so it's based on when your CD4 count gets below a certain level in the blood. You can see that that takes a long period of time. For a while, CD4 cells are low, but are not sort of all the way reduced. Then of course our patient isn't going to be completely immunocompromised. They may feel fine. Yep.
What is the seroconversion?
We'll get there. So this is from your textbook showing the number of adults and children living with AIDS in 2016. We think that there are about 37 million people in the world who are currently suffering from AIDS. And that is in addition to the 35 million who have already died. And you can see that this is a huge epidemic leading to a whole lot of sickness in a lot of people. Sometimes people think that it's not really a problem in the US or in Western countries anymore, but you can see, for example, there's 2.1 million people who are infected in those areas. It is, in fact, definitely still a real problem here. So one thing I will just mention really briefly, I know I'm going really fast, but I really like this stuff, and I want to be able to talk about as much as possible, is that I have learned over time that there's a little bit of confusion where some people think that anybody who is immunocompromised has AIDS. And that's actually not true. There are lots of reasons why your immune system can be compromised, where you can be immunosuppressed for lots of reasons. AIDS is a specific type of immunocompromise, but it's not like we don't generally use the term AIDS for like everybody who has who's immunocompromised. So like if you get a kidney transplant and go on cyclosporin, you do not now have AIDS. You just are immunosuppressed. And I've seen that confusion a surprising number of times, so I felt like I should address it. This specific immunocompromised state is caused by infection with a specific virus. This virus is called the human immunodeficiency virus, or HIV. And you can see HIV here. HIV has 9 genes, so it's causing all of those millions of deaths and all of those problems with 9 genes. It's about 10,000 base pairs long in terms of the genome. The virus looks like this cartoon that you can see here, and in fact really does look like that in these electron micrograph figures. You can see that first there's sort of those blue curlicue things in the middle. That is the genome. So there are 2 copies of an RNA genome. You can see that there are some little circles in the middle. If I currently had my retrovirus cookie cutter, I could color them with different frosting colors. It would be great. And those are really 3 enzymes that the virus has to carry in order to do its replication cycle. And I'm going to talk about each of those 3 enzymes as we go along. There also is sort of this protein shell, both the one that's shown in yellow and the one that's shown in green, that are forming the structure of the virus. And you can see, yes, it really does sort of have that conical shape. You can see the conical core here, as well as a lipid bilayer around the outside, which is shown here in purple, and a couple of envelope proteins that go through that lipid bilayer as proteins here. This one that's sort of the stalk that's in blue is called GP41. The one that's in pink, the sort of the lollipop part, is GP120. And you can see all of the genes here as well. I need to tell you a little bit about HIV replication and some aspects of the replication cycle. You'll see eventually why I had to tell you it. So the first thing that has to happen in order for this virus to replicate is that it needs to get into a cell. And that's, in fact, true of all viruses, that they need to get into some kind of cell. The way that that works for any virus is sort of a lock and key mechanism. There's some protein or some molecule on the surface of the virus that interacts with a protein or molecule on the surface of the cell and induces a change so the virus can get in. So there has to be a receptor on the cell's surface to let the virus in. HIV has two major receptors that it uses. It actually uses its main receptor or primary receptor as well as a co-receptor. The primary receptor that HIV uses is CD4. Thus, it is largely going to bind to CD4 T cells. Sometimes, for example, in my DSEM, when I'm talking about HIV, students are like, well, why don't we just not have CD4 anymore on our cells and we'll be all good? You guys, having taken immunology, can realize how CD4 did not evolve to be an HIV receptor. It has some other important functions. And so HIV has to bind, and specifically that GP120 is going to bind to CD4 in order to let the virus into cells. The virus also needs to bind to a second receptor in order to actually get into a cell. And that second receptor is a chemokine receptor, specifically some type of chemokine co-receptor. GP120 is also binding to the chemokine co-receptor. If you look in the literature, you will find a bunch of chemokine co-receptors that can work here. There are, and basically what happens is that there are different strains of the virus, and they each will use a slightly different chemokine receptor. If you were to kind of make a ranked list of all of those chemokine receptors that are being used, you would find one is used like way more than others. And then there's like a second place one, and then there's all the rest. And I don't know the numbers. I told the DSEM students that I didn't know the numbers, and I made up a number for them. And so I'll give you the number I made up for them. So 90% of the time, it uses the first place one. 8% of the time, it uses the second place one, and then every other one is sort of at the bottom. So there's like some really major ones, and then kind of you could imagine the others. The most frequently used chemokine receptor for HIV entry into cells is called CCR5. And so HIV will will use its GP120 to bind both CD4 and CCR5 to be able to enter cells. And like I said, that would be the one that I was calling like the 90% one. That's almost always used by most viruses. There has actually been a lot of data showing that for an HIV virus to be transmitted sexually, it has to use CCR5. Most of the other ones are transmitted only through like IV drug use. The other main one, or the other kind of predominant one that's used is CXCR4. That's the one that I said is like 8%. That virus usually shows up pretty late in a person's infection. And is sort of a smaller number of viruses. It's not sexually transmitted. As you can see here, this sort of oversimplifies some things that I don't want to get crazy into because I don't love the way it's been oversimplified here. But it does sort of change the cell types a little bit that the virus goes into. The only thing I will say is that usually when I say the virus goes to infect CD4 cells or cells with CD4, you're like, right, CD4 T cells, got it. It turns out that there are a fair number of macrophages that also do express CD4. And so macrophages are one of the cell types in addition to T cells that can be infected. Once the virus goes into cells, it is able to replicate and go through its replication cycle. The replication cycle is shown here. Again, there are a few specific pieces I need to hit, and I'm picking them for reasons, so I promise there's a reason. So you can see that first, the virus is going to bind to CD4 and its chemokine receptor. I don't like that chemokine receptor because it only has 6 transmembrane domains. It's supposed to have 7. And this eventually allows the lipid bilayer, the membrane of the virus, to fuse with the membrane of the cell and lets the viral components into the cell. Oversimplifying dramatically, eventually the RNA comes out. When and where, and let's not deal with. So the next thing that happens is that one of those three enzymes starts to act. That enzyme is called reverse transcriptase, or RT. Transcription normally goes from DNA to RNA. That's what you learned in Bio 250. Reverse transcriptase is an enzyme that is able to make a DNA copy of an RNA molecule. And so this HIV's genome is actually copied into DNA, and eventually we get a double-stranded DNA copy of that genome. This, of course, is the same enzyme that you used in lab for cDNA synthesis. Same process, we've just taken advantage of it for molecular biology purposes. And so reverse transcriptase is one of the special enzymes that this virus has. Most viruses do not do this. It's just a special class of viruses called retroviruses that can do this step. After the double-stranded DNA is made by reverse transcriptase, the second of the important enzymes then begins to act. This one is also really unique. One of the most common misconceptions I see with a lot of students is that this next step they think is done by all viruses. It's actually only done by retroviruses. There's something pretty similar that happens with bacteriophages, and since the kind of viruses you've learned about in your life are either HIV or bacteriophages, you decide, oh, that must be what all viruses do. And in fact, it's a special thing that only those two do. So this is not, so know that this is a special step. It's not a every virus does it step. And that is this DNA actually gets inserted into the genome of the cell. So there's this enzyme integrase that integrates the HIV DNA into the cell's DNA. That means whenever the cell replicates, it's going to copy HIV's genome too. It does not mean that now this person has HIV in every cell of their body in the genome. It's only the infected ones where the virus replicates. But if those cells replicate themselves, then HIV will be in their genome. And so you can see that here, as you can also see the integrated DNA is usually referred to as provirus. And now that integrated DNA can be transcribed, and then the mRNA can be translated, just like any other piece of DNA in your genome can. And so HIV is kind of tricking the body into copying its DNA, making its RNA, making its proteins, making all the stuff. The proteins are actually made weirdly in that they're made in these kind of multi-part proteins, which have to eventually get cut up. So here you see they're big, and here they're little. And that cutting is done by the third unique enzyme, which is called protease. It cuts up proteins. Eventually, those viral proteins all come together to build a new viral particle, which leaves. It pushes through the cell membrane as it's leaving, and it actually wraps itself in some lipid bilayer as it's heading out of the cell, where it can get some new proteins. So you will see some of the reasons why some of those steps are important and why I wanted to specifically mention them. And just to sort of elaborate on that last step a little bit, here is the process of HIV budding shown in two different images in microscopy. You can see that the virus is actually pushing out through the cell membrane and becoming a free viral particle. What I want you to notice here is that this cell is actually not lysing to just let the virus out. The virus is budding out of the cell, and the cell remains alive. And so it could make virus today and tomorrow and the next day and all the days after that. Also notice that the yellow are viruses, the blue is the cell. It's not like the cell lets out one viral particle like you've seen in the cartoons. One infected cell will make thousands or millions of new viral particles. So this is a pretty big amplification as this replication is ongoing. There are some other cell types besides macrophages and T cells that people sometimes talk about when they talk about HIV infection. I just want to briefly mention that sometimes we can see a slightly different process happening in one of those types of cells, and that is a process that's known as transinfection. And this largely happens in dendritic cells. And so what we can see is here we have an infected CD4 T cell that is making a whole bunch of virus particles, just like what you saw on the previous slides. Some of those virus particles can be bound by proteins on the surface of dendritic cells. The dendritic cells are kind of sticky and like to endocytose stuff. But the virus will not be able to actually go through its replication cycle in those dendritic cells. It won't do all the steps you saw on the previous slide. It will instead kind of hang out in the endosome or in vesicles in that dendritic cell. In some ways, it's kind of a nice little home. It's protected from antibodies. It's protected from other types of immune defenses. It's just like hanging out there. If you recall, a major job of a dendritic cell is to present antigens to T cells. And so when this dendritic cell, which has HIV just chilling in its vesicles, interacts with a T cell, it can also give that T cell the present of HIV. And so this process is known as transinfection. This cell is called infected in trans because it's not actually replicating the cell. It's just replicating the virus. It's just kind of holding onto it and protecting it and shuttling it to a T cell in order to give it to the T cell, which you can imagine is not an ideal situation for that T cell. Weirdly enough, it turns out that if you actually look at the T cells that are infected in a person, the T cells that are infected the most frequently are the ones that have the TCR to bind HIV antigens, and it's because they all got activated by a dendritic cell and transinfected by a dendritic cell, who is turning them on and also giving them HIV. So, that's cool. Because this virus is infecting CD4 T cells as well as monocytes, it will lead to the loss of CD4 T cells and eventually that phenotype of AIDS that we see in patients. But you might be slightly confused by one piece of information if you think about everything that I've told you at this point, which is that I just showed you the cell producing virus. And I showed you it wasn't lysing. It was going on to produce virus one day and the next day and the day after that. And so you might say, well, then why do the T cells die? This says they die, and you just said a couple slides ago they didn't die. So how does that go together? And there's actually been a ton of literature about why the CD4 T cells die. I will honestly tell you, if you can imagine a reason why they die, and you do a PubMed search, you will find out you are right. Because people have come up with all sorts of theories. And to be honest, probably they die for multiple reasons. It's not like they're all dying for the same reason. So one idea that people had for a while is that when the virus takes over the cell and starts replicating in the cell, that's stressful for the cell. And so eventually the cell is going to die from making those millions and millions of viral particles. And that is probably true for some cells. My PhD advisor liked to refer to that theory as, it's the virus, stupid. However, those cells that are infected with HIV those CD4 cells can also be killed by CD8 cells. That's the point of a CD8 T cell, is to kill a virus-infected cell. And so some of those CD4 cells are being actively killed by CD8s as well. My PhD advisor liked to refer to that one as, it's the immune system, stupid. We also now know that there's probably more going on there as well. So for example, We know that there are some pattern recognition receptors that can recognize HIV and lead to cell death by pyroptosis. And so that one's a little bit of it's the virus and kind of the immune system put together. But in reality, probably all of these are reasons why those infected cells are dying, but they are in fact dying. But there's one other piece to this that's really important to think about. So here you can see kind of that same process that I just showed you. We have some CD4 T cells. We can see a virus, the HIV virus, infecting the CD4 T cell. We can see that T cell might replicate. If, say, that T cell sees antigen, oh sad, it's going to replicate. And, oh wait, it's going to make virus too. All the T cells are going to die, and so we're going to end up with lots of viruses and no T cells. One thing I forgot to mention, that I'll just mention briefly for you, part of the viral genome actually includes promoter sequences to start translation and to start transcription. And it turns out that those promoter sequences are an NF-kappa B binding site, an NFAT binding site, and binding sites for all the transcription factors you've learned about all semester. So when the T cell gets turned on, oh, it turns on the virus too. The virus is kind of sneaky in that way. But this is not the only thing that will be happening with these patients' T cells. Some of those infected T cells might be memory cells or might go on to sort of develop into memory cells. If you recall, memory cells are long-lived cells. They are specifically modified to live for a long time. And so we can see those cells living for a long time. If those cells later get triggered by antigen, they can go back to being active virus producers. But while those cells are memory cells, they may not transcribe and translate the virus too much. They may be just sort of like, little Trojan horses that have the virus. They have the potential to get stimulated back and make virus, but otherwise they're just hanging out, happening to have the virus. And as a result, sometimes there is this population of cells in an infected person where we see the virus that's basically sitting in a latent form. Those cells themselves are going to replicate a bit. They're going to make virus every so often. It's sort of at a low level, this low ongoing replication. This could also happen if a memory cell itself, if the cell was already a memory cell and it got infected. And these cells make up something that is known as the HIV latent reservoir. We have a bunch of great therapies, which I may or may not have time to mention today, that kill the virus based on blocking viral enzymes. In these cells, there's not a whole heck of a lot of virus replication. And so the enzymes aren't really doing anything. And so in fact, these cells escape getting killed by the medications. Since these cells aren't really making any viral proteins, they're not making any viral antigens to get CD8 T cells to kill them. They're not feeling particularly stressed in order to lead to cell death. And so these cells are going to live for a long time in our patient, but always have the possibility of sort of going back and producing more virus. And so in fact, one of the big challenges in the HIV field right now is that we can actually get rid of HIV from patients' bodies except for the latent reservoir. We have no way to get rid of the latent reservoir. And so that means patients have to be on medicine for the rest of their lives, have to be treated for the rest of their lives, and the virus is always going to come back. We don't see any cases where the virus doesn't ever come back out of the latent reservoir. One other thing is that there may be some other places where the virus is hiding out, possibly in sites of immune privilege. But we also do know that because there are all sorts of receptors on follicular dendritic cells in the lymph node, and those FDCs are sticky, that virus usually also decorates those and gets protected just sitting on FDCs as well. And so an HIV-infected person will have a large number of these latently infected cells, giving them this latent reservoir. Did you have a question, Mark? Okay. So here you can see a picture showing you the course of HIV infection. This is the version from your textbook, but this is a pretty famous figure in the HIV field. So what you can see is that at the beginning, our person doesn't have HIV. Surprise, surprise. However, they can be infected, and their level of virus will go up to a pretty high level. You can see that their CD4 cells will crash at that point. But the virus will eventually come down to what's known as a set point level. And CD4 cells will come back a bit. And then there'll be this long period of time where there's sort of, a back and forth battle between the HIV and the CD4 cells. But what you will notice is that when the virus comes down, it doesn't go all the way to zero. It just comes down. It's always still present in that latent reservoir. And eventually, that virus is going to be able to come back out of past the control of other responses, which will further crash the CD4 cells and lead our patient into AIDS. So I want to mention, the point of this is to move us into talking about the immune responses to HIV. Because sometimes people are like, well, does the immune system do anything? Does the immune system give up? And no, the immune system actually does quite a bit in response to the virus. But I actually, in order to talk about that, want to go back to one of these past slides for a second. Yep, Mark. That's a very long answer. But the basic question, or the basic answer is, it is not clear in all of those cases whether the chemotherapy will clear the full latent reservoir. And so in some cases, because it's killing rapidly dividing cells, if those latently infected cells are not rapidly dividing, they may survive. That's the short answer. Here you can see that data I showed you on the AIDS progression and loss of CD4 positive T cells. And what I want you to notice is that you can see here AIDS. You can see that patients are asymptomatic for a while when they have low levels of, where they have reduced levels of CD4s, but still kind of enough to protect them. But this slide also shows that at the beginning, the patient is going to undergo some flu-like symptoms. Why is that patient going to have some flu-like symptoms right at the beginning of their infection? Yeah, Emily?
Because of that huge spike in the replication of the virus in the very beginning.
So there's a huge spike of replication in the virus in the beginning. Yep, that's part of it. Yeah?
Is it because there's going to be a huge response from the immune system because there's
Be more specific when you say a huge response from the immune system.
like
Inflammation, the innate immune response. This is all just innate immune response to virus. This is actually usually referred to as acute viral infection by most virologists and immunologists. Flu-like symptoms is really, that means you got some virus. And your body is responding to some virus. And so if someone is feeling kind of feverish and achy and malaise, they're like, oh, I got the flu. They don't usually say, oh my gosh, hey doc, I might have HIV. And you can also imagine that when doctors are taking history, they don't say to their patient who comes in with flu-like symptoms, could you have possibly just gotten HIV? They don't. They say, do you have a cold? Drink orange juice, right? And so, in fact, none of these diagnoses are made at that point. And that's something that, if you are thinking about medicine, something to think about. So we can also think about aspects of the adaptive immune response. In red, you can see the virus. And so again, you can see that virus peak really early and the virus coming down. You can see that it does not go to zero. that it stays at this nice low level, or not nice low level, of latent reservoir for a long period of time, and eventually comes back. What you will notice is that there are antibodies and there are CTLs that can be seen in patients in response to this virus for pretty large amounts of time. So you can see lots of different antibodies to different proteins. What you also can notice here is you can kind of see that the CD8 T cell response is happening around the same time as the virus is crashing, as the virus is coming back down, which does indicate that the CD8 T cells might actually be really important for getting the virus from that high level back to kind of control at that lower level. I will admit I worked with one of the people as a graduate student who kind of founded some of this idea, so perhaps I am biased. And so, in fact, the idea is you can see the virus replicating to a peak in pink and then being controlled. And that control seems to happen right around the time that the CTL response is really going up to a high level. If you actually take animals and get rid of their CD8 T cells using an antibody, so they have no CTL response, you can see that the virus goes up to a peak and never goes back down. And so, again, these types of things indicate that the immune system is doing a really important job in trying to block the replication of this virus. The other reason why it's important to know about the immune responses to HIV is that by and large, the way we detect HIV infection in a patient is not by looking for the virus, but it is in fact by looking to see whether a patient has developed antibodies against the virus. And so most patients are tested with either an ELISA or a Western blot to see if they have developed antibodies at a high measurable level to some viral antigen. And so you can see that one type of antibody is detectable at about 14 days after infection. Others are detectable by ELISA and Western a little bit later. And so that would be when our patient is first going to test positive for HIV. And this point is called seroconversion. The patient's serum has gone from being negative to positive. They have seroconverted in their serum. One thing that you can notice is that there are ways to use PCR in order to detect the virus a bit earlier. Those are not sort of standard lab procedures in most clinical labs, and so most patients are not originally given an HIV test with a viral measure. Sometimes it is done in patients after they've tested positive to kind of measure their prognosis, but due to sort of cost and technical limitations, that PCR is not done. Even with that PCR, you'd have to have virus at a somewhat high level before you're going to detect it. And what you can notice here is that that PCR only can detect virus at 10 days following infection. You can see that the antibody tests start being positive at about 14 days following infection. And so, in fact, we have this period of time where HIV patients actually are not detectable or cannot test or do not test positive. That's known as the eclipse phase, where we actually can't even find these patients. And so this is a serious problem, and it's where these patients are not even going to be known to be HIV positive. And you might say, well, okay, maybe that's not a big deal. But if you think about it, that's in the time period where those patients have their huge amount of virus. That's when those patients are at that high level of virus. And here you can see the probability of a patient transmitting HIV at different times. The sort of reddish-orange stuff is blood here. And so what you can see is that at the beginning, our patient has huge amounts of HIV in the blood. And you can see their chance of transmitting is pretty low. 1 out of 30 chance of transmitting. Eventually, their virus goes down to a pretty low level. And in fact, their chance of transmitting is pretty low too. It's easier to transmit when you have more virus. And then when you're at the end, it's more likely to transmit again. Unfortunately, this area where patients are able to transmit most easily because they have the largest amount of virus is before seroconversion. So patients before they know they are infected are often some of the most infectious. Also notice that in the yellow here is actually showing viral load in semen. And you can see again that there's a big change over time. And you can again see that patients are probably doing most of their transmission early before they know that they are infected. And that tells you that, you know, things that somebody might not have ever thought were all that interesting about like designing new PCR tests for HIV that could detect it earlier might actually be a big thing to help in this epidemic. That, of course, that slide mentions transmission, and there are other things I can say about transmission as well. These data show some information about the frequency of transmission with different types of activities. There are actually even more like very detailed versions of this that sometimes I show in class, and everyone feels very awkward at the things that they never expected to see in a class. But when you work on HIV, you get to learn about how to talk about all sorts of things. So if you look at these numbers, what can you observe when looking at these numbers? There are usually kind of two big things people take away from this. Yeah, Nick? Male-to-male transmission is the highest risk sexually. And so that ends up being one of the reasons why we see different frequencies in different populations. So that's a big part of it. Yep, Tom? Blood transmission is a lot higher than sexual transmission. And that actually leads us to think a little bit about the fact that the mucosal barrier, the barrier organs we talked about at the very beginning, does actually seem to do something. If you look at types of transmission that don't have a mucosal barrier, they are way more efficient than the transmission that does have a mucosal barrier. And you can see sort of that barrier here that the virus might have to make it across in some of these cases. Thinking about that, whether we want to induce immune responses at mucosal sites, thinking about kind of how we imagine this in how we model transmission in animals in different types of studies actually becomes really important. Anything else that is a take-home message from this? Yeah, Mark?
Without treatment, transfer from mother to infant is very likely.
Yes, without treatment, transfer from mother to infant is very likely. So when you look at these numbers, how do you feel, like, are they higher or lower than you think? Yeah?
I didn't expect the transfusion of infected blood to be really, really high.
Yeah, it's basically if you get infected blood, it's happening. But what about some of the other ones? Yeah.
I feel like the risk of infection is a lot lower than people think it is.
Yeah, so a lot of times people look at this and they're like, this is way less risky than I ever thought. What I will tell you is that these data are all self-report data. So these all require someone to say how many times they did what with who, and remember it perfectly and all that kind of good stuff. And also, this is a per act. So, thinking about how 100 times one does something or something like that, then you can get into some pretty appreciable numbers pretty quickly. And so this has also actually led us to thinking about how do we tell people what risk numbers mean? Because sometimes if I just tell people risk numbers, they're like, oh, that's no big deal. And actually, we then have to spend some time defining what do these numbers actually mean and what would a big deal look like with this. Yeah, Sam?
So I don't know if you know this,
Mhm.
but are you allowed to operate on people if you're HIV positive?
Yes.
What if you cut yourself?
So you use what are known as universal precautions, and so you are always there are two things. One, you're always using sort of personal protective equipment and gloves and things like that to do that. and B, which we'll see if I have time to go into this, the medications that we have reduce the amount of virus very, very low. And so likely you are probably going to be on medication if you're that surgeon, so that you have very little blood, or very little virus in your blood to be less likely to transmit as well. Okay, so at this point, you might say, okay, awesome. So our immune system makes a response to this virus. Why does it fail? We talked about the fact that it does make a response. You might have originally said that the immune system does nothing. But why does it actually fail? Why does the virus come back and actually kill the patient and not go on for a long period of time? Yeah.
It's kind of like what we went earlier over Ebola, like hiding in places, so it's hiding until it can eventually
So that
keep reappearing.
that can be part of it. There's a bunch of reasons why this eventually fails. One reason is because it's targeting the CD4 T cell. And if you remember, the CD4 T cell is helping to make many of the other types of responses work well. So if you get rid of CD4 positive T cells, then you're going to also be limiting B cells. You're going to also be limiting CD8 cells. You're going to also be limiting all the other cell types. So as you lose CD4 T cells, your immune system is going to get weaker as a whole. Many of the T cells that are responding to these viruses will undergo chronic exhaustion because they are seeing that viral infection over such a long period of time. Many of the CD8 T cells in a patient who has HIV are in fact exhausted. If you look at histology at the thymus, it turns out that HIV infection actually leads to thymus destruction. And so your thymus actually ages too fast, and you stop producing new T cells. So even if we got rid of the virus, your thymus would be in bad shape, and you wouldn't be necessarily making more T cells. So your thymus should look like this, with lots of cells and lots of development and lots of space. And it eventually looks like this in an infected person. And as your textbook likes to say, lots of other things happen in terms of the immune systems of HIV patients. One of them so this mentions things like, reduced thymus function and exhaustion and no help and all that business. The one other thing I do want to mention that's up here is that there are some big changes in the innate and inflammatory response, where patients actually are often seeing chronic inflammation because they have this chronic viral infection, which just messes up everything else. It's sort of like the T cells are both getting exhausted and getting too much cytokine. And so everything gets polarized wrong, all sorts of cells are being signaled incorrectly, and sort of all of that interconnection of the immune system that we've seen all sort of ends up being thrown off, and the patient's immune system is very largely destroyed. And so you can see that this is sort of not a great immunological outcome. But there is one other really important reason why immune control is ineffective for HIV. And this is perhaps, I think a lot of people talk about this as the most important reason. And it goes back to one of the enzymes. One could make an argument it goes back to two of them, but one of them right now, that I told you about before. And that is the enzyme reverse transcriptase, which makes DNA copies of the RNA. It's really cool. It's like this special enzyme that you don't have in your cells that can do all this fun and exciting stuff. Yay, reverse transcriptase. However, reverse transcriptase also kind of sucks at its job. In that, it makes a lot of mistakes. It makes a lot of copying errors. So if we round the genome to 10,000 base pairs, I actually never quote the exact number, because weirdly enough, the exact number is really similar to the seating capacity of the Duke basketball arena, and I always switch the two numbers in my head, and so I just say they're both 9,000 and change, and I can't remember which one's which, and so they're both just 9,000 and change. Because I went to Duke, and like our arena's a big deal, and I didn't miss a game till senior year, and like the whole thing. So like I can't remember which number's which. I just can't. So they're both 9,000 and something or 10,000. The reverse transcriptase mutation rate is 1 out of 300,000 bases, and you might kind of think that that's not too bad. But in a person who is infected with HIV, 10 billion viral particles are produced per day. And if you take that 10 billion viral particles and multiply it by the number of bases, or the seating capacity of Cameron Indoor Stadium, then you will, you find out how many new nucleotides are made every day, and then you divide that by 300,000, how many mistakes are made, you find that there are 333,333,333 mutations made per day. And if you look at that number, there's something important you should know about that number. Yes?
It's big.
It's big. It is in fact big. Can you say anything more about how big? Yes. It's bigger than the genome. What that means is that every base pair is mutated every day in an infected person. And so if we throw anything at that virus, any antibody, any T cell response, the virus makes a version of itself that can potentially evade that response every day. So this is my other favorite way of looking at this same data. Some people hate this slide, but I love this slide. So this is a phylogenetic tree. And this is, you guys know influenza. You guys knew that influenza is a really diverse virus. It's so diverse that you have to get a different flu shot every year because the virus is so diverse, right? So this is a phylogenetic tree of all influenza sequences that were known as of 1996. You might look at this and be like, that is a really ugly phylogenetic tree. Why the heck did she show us this ugly phylogenetic tree? And the reason why I showed you this very ugly phylogenetic tree is that that is HIV, one gene, 1 year, 1 country on the same scale. And so that is why the immune system is not able to control this virus. Because while the immune system can control the virus for a while, the virus always eventually finds the mutation combination it needs in order to avoid the immune system and eventually come back. And so basically the virus is in that latent reservoir mutating. Eventually it hits the right combination and is able to replicate away from the control by all of the types of immune cells. Yes, Vanessa?
How many mutations in
Mhm.
just a day?
Mm-hmm.
How does it prevent itself from basically like messing up the enzymes that it needs?
Yep. So if you look at this slide, this actually also gets at the fact that the viruses that we see around the world are actually quite diverse. So there are sort of different substrains that people have described, some of which predominate in different parts of the world. So here you can see sort of the phylogenetic tree of all the substrains. So like there's one that's type B, and type B is predominant in the United States and Western Europe, whereas type C is predominant in sub-Saharan Africa. It ends up being kind of important because you might think, okay, if we're going to come up with a treatment or a vaccine, is the vaccine going to work for all the strains, or are we going to have different vaccines? If we have to have different vaccines, which one should we make first? If the United States government is paying for it in the NIH, should it be the United States strain? If the most infections are in sub-Saharan Africa, should it be the sub-Saharan Africa strain? And so there are sort of arguments like that that are made. So Vanessa asked the question of how it is that when the virus is making this many mutations per day, it doesn't destroy itself. And in some ways, it kind of does. So where's my number slide? There. So the virus is making 333,333,330 mutations per day. You're making that many mutant forms per day. All of them are not viable. Some of them will never work and will never replicate again. But basically, you're setting up a natural selection experiment where you're making all of these mutants. Some of them are less fit and will never replicate again. Some of them are more fit and will keep replicating. And so all of them aren't going to necessarily live. But if you get one that is more fit than the original, it's going to really go to huge levels and maybe end up making huge numbers of virus. With any individual infected person, the virus is completely not homogeneous. And one of the reasons why I mention that, why I sort of bring this up, is if you look at the phylogenetic tree, you can see that there are some kind of areas where the virus has not gone. Like you could imagine a branch like here, and the virus does not seem to be able to actually go down that route. That seems to be sort of a structural constraint. And so it's not like the virus can take any old route it wants, only some options seem to be available for it. But as long as, you know, one of those is more fit than the parent, it's all good. And the reason why all this can happen is because we do have that ongoing viral replication in the latent reservoir. If we got the virus to 0 here, if we got to 0 instead of to a little bit, then it wouldn't matter that it was good at mutating because it wouldn't be there, so it couldn't come back. But the fact that we have a little bit and that little bit replicates, it basically is getting to keep trying and keep trying and keep trying to make a version that can replicate out of control. And so this virus is able to do this process because there is this latent reservoir providing an opportunity for low ongoing replication and thus low ongoing mutation. That mutation can lead to immune escape, as I have mentioned. One thing I'm just going to skip to a couple of key things now because I have other stuff I want to make sure I hit. So one of them is that there is a phenomenon known as recombination or superinfection. So we can actually see situations where a cell can be infected with 2 different genetically diverse strains at the same time. And in that cell, we can have recombination of those to make even more diverse types of HIV virus. So this is yet another mechanism of diversity. And this doesn't just happen at the level of a cell. If we have a person who is infected with one strain of HIV, like the red strain, they can in fact be reinfected with a different strain. And so you can see that this person has red HIV, can get reinfected with blue HIV, and in fact can then transmit red, blue, or new recombinant strains that formed in their body, like purple, because red and blue make purple. And so this superinfection process happens. There's a lot of variation in this process. So some people get infected and their CD4 cells crash right away. Sort of the average is that people get infected and their CD4 cells crash over about 10 years. That's kind of the typical average. There are also some people who get infected and their CD4 cells can stay high for a long time. And there are people we know of who've been infected since like the 80s whose CD4 cells are still staying high for a long time. This is all without treatment. People have looked a lot at the genetics of what could be going on here. And there are 2 genes that have really come out to be important. One of them is MHC. Big deal. The other one that's more interesting is CCR5. And so we saw CCR5 being one of those receptors that's really important for infection with this virus. It turns out that there are some people who have a deletion of 32 base pairs in CCR5, which results in the protein not going to the surface of the cell. If you have 2 copies of that mutated form of CCR5, you have no CCR5 on the surface of your cell. As a result, viruses that use CCR5 cannot infect you. And we talked about how those are the most frequent, and those are the only ones that are transmitted by sexual transmission. If you have 1 mutant copy and 1 wild-type copy, you have less CCR5 on your surface. You are actually going to have reduced viral replication. You're probably going to do better in the long run with your infection. If you have 2 good copies of CCR5, then you're going to have infection.
Does this have anything to do with malaria?
It does not have anything to do with malaria. So if you actually look in the population, the percentage of people who are homozygous for this mutation is way higher than it should be based on certain types of genetics. This mutation is actually present in a relatively large percentage of people. One of my DSEM students today was like, oh yeah, by the way, I did 23andMe and I'm at CCR5 double. Like they just, that was like what they decided to tell me today in DSEM. So there are actually a fair number of people who have this mutation. Most of them are Northern European Caucasians. And if you actually look at the frequency, the highest frequencies are in these areas that are in red in Northern Europe. The frequency decreases along this line. There are a bunch of different hypotheses about why there may have been natural selection for this. Most of them, I think, are baloney. I have 1 minute left. I gotta just go. The reason why I mention this is that there is a pretty famous case of HIV cure that does rely on CCR5. This patient had cancer and AIDS, so very bad luck. This patient was treated with radiation and chemo, kind of like what Mark was saying, and this patient got a bone marrow transplant. But when they gave that patient a bone marrow transplant, they gave him bone marrow that had the deletion in CCR5. And so the virus couldn't come back. So they gave him this kind of bone marrow. This was done in 2006. He is still off of treatment, and he has been reported as cured. This summer, this was done in a person in Berlin. He's known as the Berlin patient. This summer, a second one was done called the London patient. It's the same thing. There are still a lot of questions about this therapy. So you may have heard the CRISPR babies scandal that happened about a year ago. They were actually genetically modifying kids to try to repeat this CCR5 mutation. They messed it up, but that's what they were trying to do. There was also a paper that came out this year that said that having the CCR5 deletion led to other health effects, because ideally you'd want to know, like, do these people have any other problems, or should we block CCR5? It's not totally clear. We kind of think CCR5 might be the chemokine receptor for trafficking to the brain and GI tract. And since we don't tend to study the brain and GI tract all that often, because it's hard to pull those out of a mouse or a person, unlike some other stuff, maybe it has an important role that we don't know about. Right now, we think those patients are healthy, but we don't totally know the details right now. So that's very much a hot area of study. That's the story with that treatment that I don't really have time to go into. Yep.
Can someone still get reinfected if it's not from the CCR5?
Yes, so they could be reinfected with a viral type that does not use CCR5. So they are not completely protected. They are just sort of protected from that predominant strains. What I hope that you realize this semester is that there is a lot that we know about immunology, and it's really cool, but there are also a lot of open questions. And so, like we've done a great job of going from really broad treatments to kind of specific treatments, but there still could be better. You guys mostly came into this semester wanting to ask questions about, like, why is it that I have to wear a coat when I get sick? And, like, what food should I eat to feel good? Like, if you look back at your surveys from the beginning of the semester where you say what you wanted to learn about immunology, that tends to be the stuff you want to learn. Immunologists have done an awesome job at describing the mouse immune system. If you go back and actually look at things like when we talked about white blood cells and what % of your white blood cells are neutrophils for healthy is 40 to 70%. Like, we haven't really even, there's actually this place where we need to go back and describe healthy immune systems and actually try to come up with parameters so that we can start to answer those questions. The fact that we haven't done that means that we actually don't have any evidence when somebody says that their weird whatever drug that they're trying to sell you is an immune booster. We actually don't have enough evidence to say that it's not, and so we can't sue them and get that taken off. Which actually does happen with, like, cardiovascular stuff because they have such good evidence. So, like, Cheerios has to have, like, specific words it's allowed to say about how it helps heart health. You can say anything you want about immune boosters because we don't have that information done as well. One thing that you might notice from those papers that you read that you were supposed to read, or the choices of papers, is how many of them were actually starting to look at those types of issues, about exercise, about diet, about things like that. Like, we're finally now knowing enough that we can start to answer that question, but most of that was, done in the past year. where we're starting to answer those questions in a really kind of big picture, simple way. So hopefully we will answer those questions for you in the future, though I cannot tell you what to go eat right now. In any case, thank you guys for an awesome semester. I'm sorry I'm rushing so much today, but blame the snow on Monday. And yeah, thanks for an awesome semester. Come to the review session, and yay. Thanks. Where's Dunaway? Yay, thanks. And eat the snacks.